Good morning, everybody. So first of all, thanks a lot for coming, so many of you. It's really nice to see. And at the same time, a huge thanks to all the speakers, because it's especially thanks to them if you joined. Today is going to be quite dense, the presentation. We will start with a quick overview of the openhdemg project, about some important principles of the project. And then we will immediately go to the more, let's say, practical part. During the second part of the workshop, we will be doing a mix of theory. So we will give you basic concepts of motor unit analysis. And immediately after, we will show you the code written with openhdemg that is necessary to do this analysis. We will cover some topics that are, let's say, the major topics in motor unit analysis. Okay, so openhdemg is an open source framework. It is very important to say that it's a community-based framework. It is written in Python 3 and is mainly designed to simplify the analysis of high-density electromyography recordings. I guess you all know high-density electromyography, so what it is used. And especially for those that are a bit newer to the technique, I think you already know that there are a lot of technical difficulties in approaching this technique. One of these is for sure the signal processing complexity. With openhdemg, we want to try reducing the barriers to motor unit analysis by indeed reducing the signal processing complexity. But at the same time, we are also working on reducing the interpretation complexity. And this is mainly done via dissemination knowledge of the dissemination. The openhdemg framework is a framework. That means it is composed of different tools, different elements. One of these is the library. The library is the core engine of the openhdemg framework, and the library simply means a collection of functions that you can use while creating your own scripts. And a function is essentially some code that allows you in a few lines to do very complex operations. At the same time, we also have a graphical user interface. The graphical user interface allows you to interact with the library's function without writing code. So you press a button, the user interface is calling the function for you. But we also have a website, which is very important for the dissemination part, and it also contains all the information necessary to use the library, so to customize the various functions and to do the analysis that you want. To complete, let's say, this framework, we also have a discussion forum. The forum is hosted on GitHub, and you can, in this forum, search for support, because we know that coding is difficult, especially Python, that may be a new language for most of you. So if you need support when using the openhdemg library, you can go to the discussion forum, ask questions, or even help other people that need support. Okay, something very important for today's presentation is the difference between the graphical user interface and the library. As I already said, the library is a collection of functions. These functions can do mathematical operations, but they can also do plots, for example. And sometimes when we do analysis, it's particularly important to look at what we are doing. We will see examples later on, for example, for conduction velocity or motor unit tracking. And therefore, you need a sort of user interfaces. There are some user interfaces, the ones you see on the right, that are defined functional user interfaces. These are part of the library and can be called with functions that are written in the library. On the other side, we have the graphical user interface, which is a collection of buttons. So it looks like software. It's the green one. And with this one, we interact with the library. So one important thing to understand is that the library can do everything that can be done by the GUI, but the opposite is not true. Indeed, in the GUI, you have a big limitation, and the limitation is that you can only do what there are buttons, or what the buttons allow you to do. Today, what is my goal, what I want you to take home from today's presentation, is that using the library is really simple, is much more powerful and flexible, so whenever possible, you should prioritize the use of the library. Although I can understand that it's much more convenient and simple to use the GUI. So the openhdmg project began more than 1 year ago. 1 year ago, we released the first version of openhdmg. It was a beta release. Beta release means that it's a little tested. Indeed, we only tested it in the laboratory. And then thanks to the feedback of the users that arrived over time, we managed to solve some issues to improve the code, and most important, to add functionalities that you expressed to be particularly important for the use of the library. And a few days ago, we are very happy to say that we released the first stable non-beta release. A non-beta release means that we are now confident that all the functions are working correctly, and we are also confident that there are enough functionalities to be really useful for the user. In this time, we have not just been coding, we have also been involved in several dissemination activities. Two of them very important are the publication of the tutorial article on the Journal of Electromyography and Kinesiology, which is explaining you many important principles of motor unit analysis, and is also providing you the code necessary to do this analysis. One other interesting presentation was the participation at the Swiss Reproducibility Conference, which is a conference entirely dedicated to open and reproducible science, which fits very well with our scope. And in light of this, I leave the stage to Paul, which will talk you about the importance of a community and open science, which are core principles of the project.
All right, thanks, Giacomo. Wrong one. Imagine you're reading a paper, a very fine one, and there's some very cool code in there that you want to use, but you can't because the code is not open source, right? There's no way of you finding it. There's no GitHub repo, no everything that you can actually use. That's a bit bad, and it happens to me more often than I like. The opposite of this is adhering to open science principles. And in the next 10 minutes, I want to explain to you what open science actually is and how openhdemg fits into the big picture of it. Now, open science, I guess all of us here have heard it. Some might even know what it is, and some might oppose it completely. But my goals for the presentation are, first and foremost, for you to know what open science is, right? To also know how you can leverage it for your career, and why the community is the most important part of openhdemg. Research should be transparent, reproducible, and collaborative. That is basically the motto of open science, right? It's a movement that aims to kind of dissipate the science, the research, and the data for everyone, for a science community, but also for the general public. It therefore intends to increase the reproducibility, the transparency of research, but also tries to establish a collaborative research culture. Maybe some of you have seen those. These are open science badges. They were developed by the Center of Open Science. It's based in the USA a couple of years ago. And it's simply an incentive for authors to adhere to open science principles. The one on the right below is the Open Data badge. Simply, you put your data openly available for everybody to use it. The one on the left is the Open Materials badge. You put your materials openly available. Same thing, analysis script, statistical analysis script, or dedicated protocols. Some journals have opted to use these badges, and I think it's quite cool because if you adhere to the guidelines that these badges entail, they will be displayed next to your manuscript title. So other people can directly see to which open science principles you adhere. And I guess we all can agree on open data, open materials. But pre-registration? Well, yes, actually, and we discussed it quickly in the session before, pre-registration is a part of open science. Some people may say it kind of increases the rigidity of your protocol, but you can make changes to it. And just quickly, pre-registration is you put in a standardized form what you are intended to do in your manuscript, populations, materials, analyses, and so on and so forth. So it is very important because people can actually see what you did and try to reproduce what you did. And as you all know, we have a publication bias, so on and so forth. So this actually tries to intend to reduce this. Now that we've heard a bit of what it is, what are actually the benefits for you? Well, first of all, it's visibility, right? It's visibility of your work. If you publish your paper open access, which is sometimes hard, I know. But if you publish a preprint, or if you publish your code, your analysis script, whatever, people can see it. People can see you, and people can see how good you've done. The other thing is credibility. Right now, we are focused on credibility that is given for manuscripts, right? So you publish a paper, that's cool. Well, but manuscripts are not just manuscripts. There's a lot of work behind them, right? You collect the data, you cure the data, you write a statistical analysis script, you try to give some tutorials for other people to use it. But why don't put it open, right? You can, in your resume, then not only reference your paper, you can reference your data, you can reference your statistical analysis script, you can reference your code, whatever. So you have a lot more to show for actually of what you did. And the last thing is reproducibility or reusability, which means that, as I said before, people can go in your data, people can redo your analysis, and people can spot errors. And that's not necessarily a bad thing. If you do an error, we're just human, that's fine. But people can point you to it. And for example, if you publish a preprint, there's an error in there. Somebody will email you, hey, have you looked at it that way? It's very cool because you can correct it before you're submitting your manuscript. So you just benefit from that as well. There are some problems, however, to it. And Brian Nosek, he actually founded the Center of Open Science. He kind of summarized it very nicely in a paper, and this is 2012. There are papers saying the same thing that date back to the 80s, so it's not a new problem. But basically, our scientific reward system is based on quantity over quality. The incentives are just based on quantity of manuscripts, of citations. And open science, as some of you may know, is a lot of work. It takes time to curate your data in a way that other people can use it. It takes time to put it on a trusted repository, give it a digital object identifier, write a tutorial, make it reusable. It's a lot of time. So some PIs, actually people from the old school maybe, they push their PhD students or other students to just publish faster instead of taking one more month and doing everything right so that other people can actually use it. So we should try to shift whenever we can to going into quantity over quality. Another important thing here is when we try to publish fast, we tend to publish only the results that the journals want to see, right? These negative results are not very much liked. But if this goes on and on and on, file drawer effects happen, which means that we only report selective findings. The other thing just goes on our desk, which kind of limits the overall picture of evidence that we have in our literature and increases the type 1 errors that we see. Show of hands. Who knows this? FAIR? Nice. Okay. FAIR is the principle for research data or research software, which tries to make it, the F is for findable. So data should be findable for humans and machines alike. And we can do this by, again, putting it available openly. The A is accessible. Data should be accessible via standardized protocols. Same for software. You should write somewhere how people can access your data or your software. I is interoperable. For data, it means just specifying the exchange of metadata. For software, it means the compatibility with different softwares such as macOS, Windows, or other packages that you're using in your software. And reusable actually means licensing in terms of software and data, so people know exactly what they need to do with your data and what they can do with your data or software. And we try to adhere that with openhdemg. So for the F, findable, we put it just basically everywhere where we can find it, right? Put it on Google, writing a tutorial, publishing the paper open source, making dedicated releases, which is very important because then people can kind of reference the release that they're using of our software. For accessible, it's sometimes a bit tricky with software, but we clearly stated how people can access our software. And we also bundled it all together in a package, in a Python package to be exact. So it's very easy to install, right? You just need to download the package. Everything else is taken care of by us. Our software is trying to be interoperable in a way that we have tested it on macOS and Windows, so we know it works there. But we also specified the Python versions that it works on, and the other packages that it is compatible with. And lastly, for reusable, we're using a GPL license, and actually licensing might be the single most important thing that you should do when you're developing software. Only with a license, people know what they can do with your software. Can they use it openly? Can they sell it? Are they allowed to reuse it? Whatever. We also try to specify guidelines for people that want to contribute to the software and documentation guidelines. Communication is an important part of open science, but it's not to be confused with science communication, as we heard before from Gregory. Right? Open science tries to build a community and to communicate the results to public, and we try to do this with workshop tutorials, such as this one here, or as Giacomo mentioned at the Swiss Reproducibility Conference. We also have a newsletter where people get informed of the changes that take place in openhdemg, and we also have these online tutorials on YouTube for you to get started with using the software. However, openhdemg is not a funded project. So we rely on the community. We rely on the feedback of you guys to actually tell us what's working with the software and what's not, and what you do want to see implemented in the software. Maybe you've seen this one before. It's our roadmap, so to say, and we are currently here, which we are very happy about. First stable release, as Giacomo told you. But to get here, we kind of need a few more steps on the way. Don't worry, the core functionalities of the project will always be supported, like the UI or the library. But thanks to other people such as Drew, right, we already have contributors that help us bring the project further. So we have the engagement of the community, but we hope to keep it growing. We also hope for your feedback and new contributors. But engagement does not always mean contributing. Engagement just means using and experiencing the software. And maybe, if you feel like it, letting us know how it is. And lastly, here again is the discussion forum, just briefly mentioned, as Giacomo said before. This is basically our wiki. You can find what other people have asked, what questions have been asked, and you can rely on it to give you more information on the software. All right. I want to end the presentation by a short recap of my initial goals. You now know what open science is, essentially fair. You can leverage it to your benefit by increasing your visibility and your credibility. And lastly, openhdemg, as it is non-funded, relies on the community to kind of keep it growing. If you have any questions, don't hesitate to contact us.
Okay, fantastic. Thank you. Thanks a lot. Now we start going closer to the practical things. Before getting there, I think it's important to discuss the data structure of openhdemg. I think most of the people in electromyography are using MATLAB, and in MATLAB you have your own data structure. You can save your MATLAB file and reopen it and so on. You may store your data in other ways. For example, you could have them in a CSV file, so in Excel. Or if you use other software, you could get your EMG data in various formats. openhdemg wants to be interfaceable virtually with any data source. Obviously, this poses a lot of challenges for the functioning of the library. And this is why we need a common data structure where all the data that we import will converge. We are using Python, therefore we need to use a data structure that is compatible and well functioning in Python. In particular, we are using a Python dictionary, which is a combination of keys and values. which is very simple to read because it's humanly readable. For example, if we have a key that is called raw signal, you can easily understand that this key will contain your raw electromyographic signal, which will be a table in the particular case where every column is a channel and every row is a time instant. Now, I will not go through all the structure. You can find on our website extensive documentation of how this structure is done, and you can use this documentation to build your own function to load files in openhdemg. If you are not happy with all the functions that we already have available to interface with some other data sources. Having a common structure of the data is particularly important because in this way all the functions can operate well on your data, but it's also extremely important when you want to save your data because you can process them, but you also want to store them for future use, or simply you want to keep working on another software or on another programming language. Python has its own data structure, but as it is for MATLAB or for Excel, they work well only in the specific language. And since we want to give you complete flexibility of where you want to put your data, how you want to reuse your data, we decided to use the JSON file format. JSON stands for JavaScript Object Notation. It is lightweight. It is easy to read for humans and machines. It is simple to the list we saw before, but very, very important is language independent. That means you can create JSON files from any programming language, and you can even reopen these files in any programming language. So let's say you use MATLAB or R or other coding languages, you are able to re-import the openhdemg file in all these languages. And this is extremely important when it comes to reusability of the tools. Now, with this short introduction, we start the practical part. I will quickly talk about some basic motor unit properties like thresholds and discharge rate. I will not spend much time on this topic because I think we have much more interesting stuff going on after. But I will introduce some basic concepts that will be necessary for everybody, and I will show you for the first time the code or the structure of the code. So high-density electromyography, especially when we talk about surface electromyography, is a technique that allows us to record the so-called interference EMG signal, which is simply the summation of all the action potentials that reach the muscle. This signal is rich of information, which is, however, quite difficult to interpret as it is. This is why we have decomposition techniques that allow you to go from the raw EMG signal to the discharge activity of the motor unit or the motor neuron. If we combine these two information, we can also reconstruct the action potential of the single motor unit over space, thanks to the multiple channels of the grid. There are many decomposition techniques available. Some of them are completely automated. Some other require visual inspection, as we will see later on. But all of these techniques work with a simple principle, which is going from the interference EMG signal, detecting the action potentials, and reconstructing the pattern of discharging of the single motor unit, which we consider being also the discharge pattern of the motor neuron, because there is a one-to-one relationship between the action potential that we saw that is coming from the motor neuron and the one that we see in the muscle. So if we use techniques based on blind source separation, we usually also need to double check the result of the decomposition algorithm. So we need to perform some manual editing until we are as confident as possible with the result that we get. Obviously, before doing any analysis, you want to have your file ready. So you want to have all the motor units cleaned. You want to have removed the motor units with a low accuracy. And from now on, we will assume that all this has been done. Okay, so for the analysis part, we will assume that we have a file that has been cleaned in a proper way. Okay, when it comes to motor units properties, to discharge rate and recruitment thresholds, we can, I think the definitions are quite simple. For example, recruitment and the recruitment thresholds are the level of the reference signal at which the motor unit starts and stops discharging, while the frequency is the number of impulses that we have each second. And indeed, this is also often named pulses per second. Of course, we have a contraction. It can be a steady state, but it can be a ramp, a triangle, a trapezoid, whatever you want. And most important, we have variability. So when it comes to recruitment and the recruitment thresholds, some authors prefer, instead of using the first and last firing, prefer to take an average value of, let's say, the first 2 or 3 firings or the last 2 or 3 firings. When it comes to motor units discharge rate, you may be interested in the discharge rate at recruitment, the recruitment during the steady state, the gain in discharge rate, and so on. There are many things that you can do for different reasons. And this is why we developed the openhdmg library, having in mind the flexibility. So if you use the library, you can customize as many things as you want. You can customize how many firings to use at recruitment and the recruitment. You can select the discharge rate and so on. You can even select the measure of accuracy that you want to use, for example. Okay, so after this quick introduction, we go having a look at the code. During the looking at the code, we will see functions. These functions can have a number of parameters, and all the information on how to use the functions, how to set up the various parameters can be found on our website, specifically going to the API section, API reference section. There are nice and well-reading tables, even if you can see the documentation also from the code itself. Okay, we open Visual Studio Code. I think you should be already familiar with it if you worked before with openhdmg or Python, or if you downloaded the scripts that we provided you with. You can even try to run them in real time now, where you should have everything available. I remember you, if you encounter issues, and there may be many, you can use the discussion forum, and we can help you through the various steps. Okay, so we make it a bit bigger. I think it's more or less readable. As you can see, the code is really short, okay? There are very few lines of code. And you will see that with these few lines, we will be able to estimate a large number of motor units properties. First line is a comment, so it doesn't even matter as a line. And then we import the libraries that we need, the modules that we need. In this case, we simply need the openhdmg library. And we import it with the short name emg because we need to access it very often in the code. Then immediately below, we go to load the file, the decomposed and cleaned file. In this case, it is stored in JSON format, as we saw before, and therefore we are opening it with the function emg from JSON. If you don't know what importing a library means, what a function means, so if you essentially don't know Python or even coding at all, we have a YouTube channel, and we also did some introductory coding classes that you can use to learn the basics that is needed to use openhdmg, but in general to use Python for data analysis. Then we do some processing that is not even strictly necessary, which is sorting the motor units based on their recruitment order. Then we plot the motor units so we can visualize the discharge pattern of all of them. And then we use the function basic motor unit properties. This function is estimating all the commonly used motor unit properties in a trapezoidal contraction, which is very common. Then we display the results in the terminal, and we also save them in a CSV file. And as you can see here, we used 25 lines, including comments, including spaces, and also formatting of the code. So 3 lines are essentially 1. which means using the library is potentially really simple. It doesn't take to write a huge amount of code, and this is the advantage of using functions. So if we simply press play, the script starts loading the file. And as you can see here, you can visualize the discharge pattern of each motor unit. And then we close the figure, so we continue executing the script. We are requested to select the start and end of the steady state, because we also want to estimate properties at the steady state stage. Then the script asks us to insert the maximum voluntary contraction, so we can estimate both absolute and relative thresholds, and we get all the results. Now, it took very little. We have all the results here in the terminal, which are quite difficult to read. So we can even open the CSV file we saved. And as you can see here, with a few lines of code, we estimated a huge amount of motor unit properties. Okay, we have, for example, the accuracy, that in this case is the silhouette score. We have the absolute recruitment and the recruitment threshold. We have discharge rate at recruitment. We have the coefficient of variation of the discharge rate at the steady state. And we even have the coefficient of variation of the force during the steady state. So there is a number of output that are usually, let's say, the most used ones. But this is like the very basic use of the library. For example, here is if you move your mouse over the name of the function, you can see all the parameters that the function can take. For example, we can specify how many firings we want to use at recruitment and the recruitment. We can specify when the steady state starts and ends, and this is particularly important for automation of the code. Or we can specify which accuracy measure we want to use. And something that some people may find interesting is also the discharge rate range. So for example, you may want to exclude firings above a certain frequency or below a certain frequency, because they are probably an error that came during the cleaning of the motor units or the decomposition. So we cannot do all the examples now, but what is important to understand is the flexibility of the library, but it's also the simplicity of the library, because we wrote a lot of code, but you can essentially do all what we did now, so get the results of your motor units with that much code. So this is all the code you need to estimate motor units properties, and this is why the library is powerful, is flexible, and simple to use. And this is why you should try to learn it, because it's simple. Okay? So we now continue with the presentation. We go to some topics that are definitely more interesting and challenging. So I leave the stage to Andrea. And we talk about conduction work.
Okay, thank you everyone for being here today, and thank you Paul and Giacomo for having involved me in this open source project since the beginning. In the next 20 minutes, I will show you how we can estimate non-invasively conduction velocity through openhdemg. Briefly, this is the outline of my presentation. We'll see the definition of conduction velocity and the difference between muscle fiber and motor unit conduction velocities. We'll discuss briefly about the relevance in neuromuscular physiology, and I would also add in clinical research settings. We'll see which are the basic experimental requirements, mainly related to electrode placement, that are needed to ensure an accurate and reliable estimation of conduction velocity. And lastly, we'll go through a step-by-step guide about how to estimate conduction velocity with openhdemg itself. Firstly, although they measure the same thing, which is the speed of propagation of action potentials, muscle fiber and motor unit conduction velocity are two related, although different concepts. In particular, muscle fiber conduction velocity is a global EMG-derived parameter that provides us an estimate of the average or the weighted propagation velocity of several concurrently active motor units. And it can be measured in a relatively simple manner during voluntary contractions, as you can see here for a ramp, by placing a grid of electrodes oriented with the anatomical direction of muscle fascicles, and by observing the propagation of action potentials from an innervation zone to along the electrodes, the electrode columns. And it can be estimated by dividing the distance between recording electrodes for the time of propagation of action potentials on that distance, which is known. On the other hand, motor unit conduction velocity is an EMG decomposition derived parameter, so not a global EMG derived parameter, which reflects the average propagation of velocity of action potentials along muscle fibers innervated by individual motor neurons. And from an experimental standpoint, the methodology is exactly the same. In this case, it's shown an example for the biceps brachii. We position a grid, a bidimensional grid of electrodes on the muscle belly. We record the HDMG signals, and then we add an algebraic procedure that we all know, which is surface EMG decomposition. This is done to obtain individual motor unit pulse trains, and then with an additional algebraic procedure, which we'll see in a second, but I guess most of you already are familiar with the spike triggered averaging, we are able to identify and to study the contribution, and in this case, the peripheral properties of individual motor units, rather than average values of several concurrently active motor units. What's the importance in neuromuscular physiology? What does conduction velocity tell us? Firstly, it relates to the electrophysiological properties of the sarcolemma. So it provides us an indirect window into the electrophysiological properties of muscle fiber membrane, since it's influenced by mainly by ionic concentration of sodium potassium, by the activity of the sodium potassium ATPase pump, and by membrane properties such as its potential resistance and capacitance. Maybe most importantly, because conduction velocity linearly increases with voluntary force, and this is due to the linear association with motor unit recruitment threshold, in turn due to a direct biophysical association with muscle fiber diameter, which has been initially observed in animal models, confirmed through simulation, and only recently, at least is valid for biceps brachii, also displayed and found in humans. So for these reasons, conduction velocity is considered a robust size principle parameter and a strong biomarker of motor unit recruitments. This means that it can be used to assess changes as a consequence of different training interventions with respect to aging, disease, injury, and immobilization. We have to be aware that, as Giacomo said, conduction velocity is a complex task. Estimating conduction velocity is a complex task that starts from an experimental point of view. So we need to satisfy at least a couple of conditions related to the experimental part before moving to the analysis. And these are the location of an innervation zone, which can be done more directly through electrical stimulation of the muscle, or by asking the participants to produce low force contractions and visually inspect the recorded signals with a dry or non-adhesive linear array. And this is what we would need to see. So we will need to identify the innervation zone. How do we define it? As the point of inversion in the propagation direction of action potentials. This is essential to position our electrode grid. The second one is the detection of motor unit action potentials propagating, as we can see here, highlighted with the red arrows, in the direction of muscle fascicles from the innervation zone to the tendon regions. In order to do that, we can also, it is recommended at least to use a dry linear array prior to attaching the high density grid of electrodes. In this case, we can adjust slightly our array in order to observe the clearest propagation of action potentials along the channel with minimal change in shape. How do we estimate conduction velocity? 4 basic steps. I will not cover the first one because Giacomo has already discussed about the composition and visual inspection. So let's imagine we have recorded our surface EMG signal, and we have run the decomposition, and we have visually inspected the results according to the guidelines. Next 3 steps are estimation of multi-channel motor unit action potentials, and this is done via STA, or spike triggered averaging. Next one is the channel selection, and the last point is the estimation itself of conduction velocity via maximum likelihood estimation method. So now we'll cover point from 2 to 4. So step number 2 is to estimate multi-channel 2-dimensional motor unit action potentials with the STA. So firstly, we convert our monopolar recordings into double differential signals along the direction of muscle fibers. And the STA involves the selection of a specific time window in double differential channels, generally comprised between 30 and 50 milliseconds, which is centered on individual motor unit firings, and then averaging all the double differential signals within this window. And this allows us to get an enhanced representation of the average motor unit action potentials. And this is done for each channel of our grids and for each motor unit. Here we can see a comparison between the STA previously applied on monopolar recordings and on double differential signals. So the point of this partial filtering is to reduce the amount of non-propagating components and to attenuate the end of fiber effect. Thus, we get an enhanced representation of motor unit action potential propagation. Step number 3 is maybe one of the most challenging. This refers to the double differential channel selection, which is a visual or a manual selection process. From now on, I will use mainly screenshots taken from the openhdemg interface, so as you can get more familiarized with what you will see while using the app. This is a representation of multi-channel motor unit action potentials. And what we have to do here is to select visually the highest number of adjacent channels. And it's recommended to select at least 3 consecutive within the same column of electrodes based on the following 2 criteria. The first one is to observe or to look for the clearest propagation of motor unit action potentials with minimal change in shape across adjacent electrodes. And the second one, which strongly supports the first one, is to look for the highest cross-correlation coefficient between consecutive channel pairs. Where do we start? The easiest part, which is not actually the easiest, but a good starting point, is to try to locate the innervation zone. One more time, this refers to the inversion in the propagation direction of action potentials. As we can see here, the innervation zone is located at the top of this grid, and it is highlighted in red, but we can somehow see the bidirectional propagation of action potentials, which mostly propagate towards, in this case, caudal regions. Then we try to look for the highest number of double differential channels supported by the highest cross-correlation value. In this case, we would select column number 3, channels from 4 to 6, characterized by an average cross-correlation of 0.96, which is very high. Generally, a threshold of 0.7, 0.8 is recommended, and we will get an estimate of 4.6 meters per second. What happens if we don't know which column to select? In this case, we can clearly see a good propagation from channels in column number 2 and column number 3. Well, we have to be aware that the same number of selected channels, but from different columns, different and adjacent columns, may provide slightly different estimates. So both cases, we have selected 6 channels from adjacent columns. And we get an average value of conduction velocity of 4.46 versus a value of 4.56. So slightly different. What do we do here? Here, in this case, we select the column and number of channels with the highest cross-correlation coefficient. So in this case, it would be channels from the 6 channels from column number 3. Also, it is important to note that the number of selected channel influences conduction velocity estimation, and this is very important. Let's see what happens if for the same motor unit, we simply alter the number of channels over which we can estimate conduction velocity. Here we have 3, average cross correlation very high, 0.97, 4.5 meters per second. With 4, still very good average cross correlation values, slightly higher values of conduction velocity estimate. With 5 channels, conduction velocity increases. With 6 channels, one more time, similar results to those of 5, but still a very good and very high and acceptable average cross correlation coefficient. So in this case, what should we do? We should always look for the highest number of adjacent double differential signals and characterized or supported by the highest average cross correlation. So also in this case, which is exactly the same example I've done before, we would go for column number 3 channels from 4 till 9. Last step is the estimation of conduction velocity itself, which is done via maximum likelihood estimation method. First of all, we have to be aware that several methods for estimating conduction velocity have been developed over the last 30 years from surface EMG recordings. The one we have implemented here is the one that is at present the most accurate, reliable, valid, and reproducible, which is the one based on MLE method, which very briefly estimates the time delay in unidirectional propagation of average motor unit action potentials over a number of double differential channels. There is no time to cover the technicalities of the implementation of this method, which is very complex, and but I invite you to go and read the paper, the first paper that have introduced the method. I'd like to conclude with a couple of remarks. One more time, conduction velocity is conduction velocity estimation requires caution because is a variable that can be easily influenced by how do we position the electrodes and how do we select the channel among the others or above the others. We should always discard conduction velocity estimates that range outside the physiological range, which is generally comprised between 2 and 8 meters per second, since this bias may be due to errors both in data collection or in the analysis. In simpler terms, wrong channel selection. And last point, very important, is I invite you to try to be consistent with the channel selection, since the number of channels we select for conduction velocity estimation significantly influences its estimate. So, and this is valid within subject as well as when we need to assess the effect of a longitudinal intervention. So when we repeat yes, we have to handle different experimental conditions. Let's move to the hands-on part, which will be much faster. Cosa? Okay, also in this case, the code has been written very easily and in a simplified version by Giacomo and Paul. So basically, a few lines of code are needed to import all the necessary library with the functions needed to estimate conduction velocity. Then we lines 5 from 5 to 8 are basically where we specify the path and the name of the file that we have to upload and use for our analysis. Once we have uploaded our file, we go through filtering the recorded EMG signal, and next step, line 13, is that maybe may need a little more attention, is the specification of the order of channels. This, of course, is a variable that depends on the grid you have used and on the configuration of electrodes you have used. So you should look for these specifications according to the grid you've used and order them into these lines. Thereafter, we go through the sorting of the channels, and lastly, which is the most important part of the code, we run the code for conduction velocity estimation. Here we can specify a couple of variables, which are the first one, the number of firings that can be used for the spike triggered averaging. This is generally is recommended to keep a number of firing comprised between 30 and 50. And similarly, we can specify the time window for the STA. And also in this case, the time windows can range between 30 and 50 milliseconds. Once we have specified all these details, we can simply click on play. The code is running and we'll get this beautiful interface, which is exactly what I've shown in my presentation. On the left here, we can select the number of motor units. In this case, motor unit number 0. We see the two-dimensional multi-channel representation of each motor unit action potential. And then we can select the specific column for our estimation, let's say column number 3. And we look for the best propagating action potentials and along the channels of the grid. So let's say that our innervation zone here is somewhere below row number 1 or 2. let's say we select from channel number 3 to channel number 10, which are all characterized by a very good and very high cross-correlation value. And then we simply click on estimates. And here on the right, as you can see, we'll get an immediate estimation of conduction velocity and the average cross-correlation value of the channel that we have used to estimate, to make our estimation. I'm running out of time?
No, it's okay.
Okay, let's see another example. If I will remember, this motor unit was a little bit more complicated. So motor unit number 1. So in this case, at first sight, we would need to identify an innervation zone, which could be somewhere in around this area. Do we see a clear propagation of action potentials? It's a little bit less clear than the previous example, but let's go for this one. We see some action potential, this propagating in this direction, but let's see what happens if we select from, I need to move this, yes, slightly. So let's see column number 2, channels from 1 to 3. So apparently very good average cross correlation, 0.89, 0.87 and 0.9. So well above the threshold. Let's see what happens. Estimate. Can you read the value? It's probably too small. I'll read it for you. 1.78 meters per second, which of course ranges outside what is the physiological range to 8 meters per second. So in this case, we could either discard this motor unit or look for other channels handling more clear propagation. So in this case, we could go for column number 2. So same column. Let's see here. Channels from 7 to 10. 7 to 10, we see this action potential. We see in some way a propagation from this channel to this one, to this one, and this one. So we would select column numbers 2 from electrode 7 to electrode 10. And 4.88, which falls within our physiological range. As I said, channel selection is maybe the most challenging part because we need to see and observe many waveforms and signals in order to get familiar with them, in order to perfectly or instantly identify an innervation zone. So it takes time. But I think that Giacomo and Paul have done an excellent job in simplifying as much as possible, at least the part of the signal analysis in openhdemg. So with having said that, I think
Also show a terrible example.
a terrible example. Okay, yes, I thought I was out of time. Because not all motor units are as motor unit number 0. This could be a perfect example. Let's see a terrible one. Should be, no, this is also good. This one is a terrible example. What do we see here? We don't see any clear propagation. We don't see any clear motor unit action potential waveform. So in this case, and this is also highlighted, maybe I haven't mentioned that the number you see here are the cross-correlation values calculated on consecutive pairs of channels are displayed in red, the cross-correlation values that are below the recommended threshold, which is in this case, 0.8. So in most of these cases, we see a red flag. Plus, we don't see any good propagation. So in this case, it's only a suggestion, but I would discard this motor unit. I would not look for any channel in order to calculate conduction velocity. This is simply too noisy, and it might bias the final estimate. Good example, so and so, and wrong example. So perfect. Having said that, I thank you again, the audience, for being here, and Paul, Giacomo, and all the other contributors.
Thanks a lot, Andrea. Now, I didn't know there was a coffee break, so I didn't even consider it in the timing. But since we have been fast, I think we can go 5 minutes outside here. You can take a coffee. And when we come back, we have Drew that will present us persistent work currents.
Great. Well, first, thanks, Giacomo and Paul, for including me on this. I think it's a really cool thing and a worthwhile effort. So today I'm going to be
on the screen
talking a bit about persistent network currents. A fair number of you probably already know about persistent network currents a little bit. What do you want me to start this? But persistent network currents, for those maybe unfamiliar, are intrinsic properties of the motor neuron that serve as more of a modulatory role on motor neuron excitability. And so quite broadly speaking here, it's reason that all output from spinal motor neurons can be comprised of 3 primary input commands to those motor neurons. Those are excitatory synaptic inputs that depolarize the motor neuron, cause it to fire, inhibitory inputs that do the opposite, and neuromodulatory inputs here, which are where these PICs are housed, that change how this motor neuron responds to this excitation or inhibition. And so these neuromodulatory commands here exert profound effects on motor neurons and are primarily mediated by monoamines from the brainstem, such as norepinephrine or serotonin. What these monoamines do, I mean, they do multiple very important things, but one particularly important for this talk thing that they do is they facilitate persistent inward currents, or PICs, meaning they make them larger. What PICs are an intrinsic source of current in the motor neuron that both amplifies and prolongs any excitatory synaptic input. And so these PICs are mediated by voltage-sensitive channels on the dendrite of this motor neuron, such that as an excitatory input comes into the motor neuron, depolarizes the motor neuron, and causes these voltage-sensitive channels here to open up. And when they open up, they allow an additional rush of excitatory current to come into this motor neuron that amplifies the original excitatory command. But in addition to this, these channels that mediate PICs here are also persistent in nature, meaning they like to stay open. And so as this excitatory input here decreases, these channels like to stay open and provide an additional source of current to this motor neuron that prolongs its firing. And so a great traditional or classical example of this can be seen here on the bottom. Here I'm showing intracellular recordings of cat spinal motor neurons shown in response to a black current injection ramp. So you can think of this black ramp here as this excitatory input. And what you can see is that under low levels of monoamines here in pink, you can see that the discharge of this motor neuron is quite linear and tracks this input very well. Whereas under higher levels of monoamines here, again, monoamines make these persistent inward currents larger. These PICs here introduce these nonlinearities, where you have this amplification here at the onset, so that additional rush of current coming in, and this prolongation here, where this motor neuron fires at a much lower level of excitatory input than was even originally required for it to be recruited and turned on. And that's because that additional current from PICs maintaining that discharge. And so these 2 hallmarks of PICs are thought to be very critical for everyday motor tasks. It's thought that this amplification here could be quite useful for fast or strong contractions, whereas this prolongation here is likely very useful for postural control or long stabilizing holds, things like that. But in addition to it being quite critical, changes in PICs have also been implicated in various neural injury states, neural pathologies, in the aging process, and potentially even different between the biological sexes. And so there's a large group of really great people doing lots of work in this field. I purposely left off citations from the slide because there's a lot and I'm sure I would miss someone, but I'm sure if you look on the app for the conference, there's lots of PIC talks, lots of PIC posters, and I'm sure you can find some really cool work, or if you can't, reach out and I'm happy to point you to people that are doing this. But through just seeing how they could be implicated in these various states, and then also how they're potentially quite critical for normal motor tasks, you can begin to appreciate how quantifying them could be quite insightful and give you insights into intrinsic properties of motor neurons. And so to do that, there's two primary approaches, either a paired motor unit analysis methods or single motor unit analysis methods that more or less, for the purposes of this talk, align with that, quantifying that prolongation from PICs or the amplification from PICs. For today, I'm going to focus on a metric called delta F, which is more or less looking at that prolongation, as that's what we currently have implemented in the newest release of openhdemg. But we have some various other approaches that may be forthcoming and in additional future releases. So delta F. Delta F, as I mentioned, is more or less quantifying that prolongation effect from PICs. It's slightly more nuanced than that, but that's a general idea. Delta F was first introduced by Monica Gorosini in the early 2000s and is simply quantified as the change in discharge rate of a lower threshold reporter motor unit at the onset and offset, or recruitment and de-recruitment, of a higher threshold test motor unit. And so this metric is based on the premise or idea that this lower threshold reporter motor unit here is an effective proxy for the net excitatory synaptic input to the test motor unit, such that if you take the difference from when it turns on to when it turns off, this change in frequency, or delta F, is roughly equivalent or approximates the additional current that those PIC channels are supplying to that motor neuron. And so to be, I guess, crystal clear and drive home the point here, what that's implying, we're assuming that this lower threshold reporter motor unit here is an effective correlate for this black trace here, such that if we take from a higher threshold unit, if we take from when it turns on to when it turns off, that should approximate that additional current from PICs. And so for this to hold true, though, there's various inclusion, exclusion criteria for that to be the case. I'll briefly mention them when we get to the coding section again, but one very important thing to mention here is recruitment time difference between the reporter and the test motor unit. We want to make sure that when we're doing this, for this premise to hold, we want to make sure that this lower threshold reporter motor unit, that its PIC is fully active and saturated, and that the motor unit is only responding ideally to that excitatory synaptic input. And so the reason being here is that if you look at this blue trace up here on the top left, you can see that when the PIC is being activated, there's this quite high nonlinearity here. Whereas once the PIC is fully active, this motor neuron here, the discharge rate, approximates this black trace much closer. You can see how the slopes here are quite similar. And so to do that, we look at recruitment time difference between the reporter and the test motor unit, and make sure that there's a sufficient number of time. Additionally, we want to make sure that this reporter motor unit is actually responding to excitatory synaptic input. And so to do that, we make sure that this reporter motor unit is modulating its discharge rate sufficiently during these ramp contractions. And then additionally, another inclusion criteria is we want to make sure that this test motor unit and reporter motor unit are receiving roughly the same synaptic input. And so to do that, we look at the cross correlation between the discharge of these motor units, assuming that if the cross correlation is high, they're likely receiving very similar inputs. And so here programmatically, to quantify delta F, there's a couple different approaches out there. They all generally start with the same first step. So you would have a trial. Here's your trial. You have your instantaneous estimates of discharge. You can see here. And the first step would be to quantify smooth continuous estimates of discharge rate. So we need smooth continuous discharge rates so that we can look at the difference between the discharge at test unit recruitment and de-recruitment. There's various approaches to create these smooth estimates. Lots of alternatives. What we suggest and what we've implemented in the openhdemg framework is support vector regression, or SVR. If you're interested in any of the alternatives, reach out or get on the discussion board, and I'd be happy to talk at length of why we chose this. But that is what we chose. So to do delta F, once you have these smooth estimates though, we'll do this for mode unit 3 here as an example. So for mode unit 3, what we would first do is find all suitable lower threshold reporter mode units. In this very simple example, that's just mode unit 1 and 2. And then we would quantify the change in discharge between this mode unit 3's recruitment and de-recruitment for both mode unit 1 and mode unit 2. And so that will yield 6.1 here for mode unit 2 and 4.3 for mode unit 1. We will then do the same exact thing here for all possible test and reporter mode units across the entire file. And in the openhdemg framework, we've supplied 2 ways to output the data. The first is to just output all possible pairs across all test reporter mode units. And the second, which is currently the default in the code, is to take the test unit average, which is for each test mode unit. So for mode unit 3 here, we would take the average of all delta F values for lower threshold reporter mode units. So here, that would be the average of 6.1, 4.3. Quick math, that's 5.2. And so there are some additional methods that we may start implementing if there's requests in future releases, but this is currently the 2 methods that we have implemented. And so with this, you can then see how we can begin to kind of piece this together, and you could then do this across many different files, and you would get populations of delta F values for your data. So with that, I know it's very brief, very high level PIC stuff. If you have any questions in the discussion section, you can answer questions, or I'd be happy to meet with you at any time. But for the coding overview, which I imagine most of you are more interested in seeing, what I'll first do is outline the functions that we've developed, their inputs, their outputs, and then I will then go through a script where we'll just show the functions in working and the outputs. So to get your delta F values, we have to first quantify those SVR fits. So to quantify SVR fits, I have developed this function called compute SVR. It takes various inputs here. The only required input for this is the EMG file from the openhdemg framework. And then putting this in, it'll output the SVR fits. There's various other inclusion or input options here. I'll go through a couple. They will all default to a reasonable value though. So the first is gamma n, which is the kernel scaling coefficient for SVR. If you're not familiar with SVR, you don't need to have an in-depth knowledge of support vector machines to really grasp this or implement this function. But on a high level, what SVR is doing is fitting your data in a high dimensional space with an acceptable margin of error. And so what we can do is we can adjust that dimensionality expansion and various other aspects of that. But so what gamma n will do is it scales your kernel, where if you put a smaller value for this, it'll make smoother fits to your data. Additionally, we can adjust a regularization parameter here, which is just a penalty for observations of data that are outside that acceptable margin of error in the high dimensional space. So if you make this larger, it'll try to fit all of the data points and be a much more variable fit. Additionally, here you can change this, it's called discontinuous firing duration. This is the length of time that you deem discontinuity in the firing of your motor unit. So it'll default to 1 second, meaning that if your motor unit stops firing for more than 1 second, we'll fit it with separate functions. And so all of these have default values. We do suggest that you use the default values. There's a reason we chose them. So there's a couple of years ago, we published a paper detailing why we chose the parameters and what it means. So we do suggest that you use these defaults. If you do deviate from them, there may be instances where that's necessary. Just cite the paper and then maybe, cite the framework and then give reasons why you changed and what you changed. Once you put these in, this will output the SVR fits. So this will be a data frame. You'll have the SVR fits, their corresponding time vector, and then a vector here or a variable here called gen SVR, which are the general SVR fits, which are all the SVRs just aligned in a similar time vector, which are quite useful for quantification stuff. With these SVRs, you can then throw them into this function called compute delta F, which will quantify delta F. So this only requires the EMG file and the smooth fits. It'll need that gen SVR variable though. And then you can change the average method as an input as well. So you can either use test unit average or all, and it'll output whatever you choose. You can also choose to normalize your delta F. Currently, this will default to false. We suggest you use false unless you have a in-depth understanding of why you would normalize. If you think you need to normalize, I'd be happy to talk about reasons where that's applicable. Also, you can change all of these different inclusion-exclusion criteria. So they will default to a value, a reasonable value. And if for whatever reason you have reason to change them, you can. This is a recruitment time difference. That's the time between reporter and test recruitment, cross-correlation cutoff, and the control unit modulation here, which is that modulation depth of that reporter motor unit. Also here you can pass in a Boolean clean as either true, where it'll take all those, remove the units that don't meet that, or false, where it will ignore those. This will output a data frame with 2 columns, motor unit. So if you've chosen the all method here, it'll output the pairs that created that value. If you've chosen the test unit average, it'll output the test unit that created the test unit average. And then DF is just the delta F. And so for the hands-on, I don't know if everyone's following along or if you're just watching along. Either way. In the file for the workshop, there's a pic.py. This has all the functions I mentioned. So what we'll do is we'll quickly run through this. I will probably run through this in debug mode and have breakpoints so that we can stop along the way. But let's run this. So what this script will first do is it'll import the library. It'll then load an EMG sample file. It'll then sort the motor units. I find that sorting the motor units is useful for visualization, but also for delta F calculation or interpretability of them. It'll then compute the SVR with the standard default parameters. So again, all you pass in is the EMG file. We'll then extract those gen SVR fits, and we'll put them into this function that Giacomo made, where it'll plot these in a very nice way. So we'll throw in the EMG file, we'll throw in those gen SVR fits. We will then throw in a couple other input parameters. So we want to see all the motor units. We want to see the instantaneous estimates of discharge rate, and we want to stack them in the y-axis and separate them. And then we want to also add the torque or the reference signal. So running that, you can see the picture or the figure already popped up. So this is what this will output. Here you can see the SVR fits. So here are 5 motor units. They're just offset in the y-axis. And you can see the darker lines are the SVR fits. You can see that they follow the data quite closely. These are with the default parameters, which is meant to replicate a 2-second handing window with just a closer fit on the end conditions. And you can also see here that since that firing duration discontinuity is set to 1 second, there is a point here in the green motor unit that it stops firing for greater than 1 second. So it did fit that with separate functions. Again, though, you can change this if that's not something that you want to happen. Next, what we'll do is perhaps maybe you have a condition where you want tighter SVR fits. I know 400 millisecond handings are also very popular in literature. So to replicate something more like that, what you can do is with this compute SVR, we can then change that regularization parameter. So if we make it larger, again, it's going to try to fit more of the data points into that region. So what we'll do with this is we'll throw the EMG file into the function, up the regularization parameter, and then plot it in the very same way. Cool. All right, so here you can see that it's much closer to the data, and meaning it captures a lot more of the variability in the data itself. And I did save this at the start of this. Okay, so you can see that this was the original fit, and then here on the left we have the new fit with the higher regularization parameters. So you can see it captures a lot more of the variability in the data. Again, more replicating like a 400 millisecond handing. Additionally, you can go the opposite way if that was something that you were interested in doing. Here we can make the regularization parameter smaller, so it'll do the opposite. We'll just quickly plot that so people can see. So here you can see that it's quite flat. This is not something you probably want to use, right? It's barely capturing the basic essence of it, but it is, in some instances, maybe something you're interested in doing. With this SVR, we can then begin to calculate delta F. So to calculate delta F, well, actually, those are all trapezoids, right? We'll use triangular contractions, which are more standard in the delta F quantification. To do that, there is a file here uploaded in the workshop, the triangular 20. So first we'll load that with the file path. We'll then sort the motor units, and then we'll compute SVR with the standard default methods. I'll then plot these, but this time won't stack them. So we'll just leave them on top of each other, and then we can calculate delta F. So here you can see all the motor units just on top of each other for this triangular contraction. We could then pass those SVR fits, the gen SVR version, into the delta F function. We'll first do the all pairs, and then I'll just print that to the terminal to observe the values. Right. So you can see on the terminal, it's output 120 rows. So there's 120 possible reporter and test motor unit pairs. And the column here for motor unit is the pair of the motor unit that made that delta F, and that's corresponding to the condition or the placement in the file, so the file order of the motor units. We can then also do this with the test unit average, and that should give us 15-ish values for the number of motor units in the file. Oh, yep, so 16. So this will output each delta F for the 16 motor units. And you'll notice that since these are ordered by recruitment, the first 3 motor units don't have a value. And that's because they don't have a sufficient reporter motor units to report delta F. Here you can also then output the average of all of these for the file if that's something you're interested in doing. And then the average. And so once you get familiar with this, I think it's quite easy to see how in very little lines of code, like there was a lot more code that wasn't necessary there, the plotting and everything. If you just wanted to quantify delta F, one function, output that, and you can loop it across data and then output it to CSV or whatever you wanted to get your whole study, metrics for your whole study. So with that, thank you. Anything else?
Okay, thanks a lot, Drew. Thanks also for pointing out that functions can be complex, but we provide default values that are reasonable also for if you don't have a full understanding of all the math behind. So now we go to the last presentation, which is done by Eduardo on motor unit tracking. After this, we will say a couple of more things, and then we start the question and answer session. So hold on, we are almost done. Thanks, Eduardo.
Hello, guys. Well, considering the time, I will be brief, much briefer than previous presentations, but I hope that you find this useful nevertheless. So when we decompose motor units, we need to first understand the basics. So why are we using high density CMG? And so we go back into the literature and we find this seminal paper from Farina and colleagues in 2008, where you can find, for example, here, that there are two, do we have a pointer here? It's not working. Okay, so here we have 2 intramuscular motor units, identified, of course, with intramuscular electrodes. And then we try to see the representation on the surface, okay? If we just use a single bipolar channel, you will see that even though these are 2 different motor units, that you can clearly see the actual potentials are very different on the inside the muscle, their superficial representation is very similar if we just take one channel. But as soon as we start taking the spatial profile, the 2-dimensional spatial profile of these motor units, you can clearly see that these action potentials belong to 2 different motor units. And that's, of course, are the principles on how then this can be separated with blind source separation methods and so forth. But this uniqueness, okay, that each of these action potentials have for these model units can be then used, of course, for tracking these model units across sessions or within a session as well. So for this reason, we first wanted to test whether this worked. Of course, we performed some after positioning the grid in a similar location across different sessions and also within the session, of course. We checked if these action potential profiles, okay, will be similar if we track them across time, okay? And since if I show again, this previous slide just contains roughly about a 6x6 matrix, okay? So it's not, sorry, 25 channels here. That even when you don't have a lot of channels here, you still can identify the uniqueness of these action potentials. But since you have many more channels now, so we normally use 64 channels, or the lowest usually is 32 channels, we can exploit that and really check the unique features or the templates that we get from these model units. And in this study, we could observe that these 3 model units could be matched across 3 different sessions that happened across different weeks, okay? And then if we check the firing profiles or the firing properties, their discharge times, we can see that also the recruitment thresholds match very nicely across sessions, considering, of course, that there was no intervention involved and there was no changes in of course, neuromuscular activity. By doing this method, so what's the advantage of doing this tracking? Because why do we bother to do this analysis? It's because the conventional analysis to assess model units, for example, for an intervention or across session was yes, you identify your model units and then you average, for example, their model unit properties, such as discharge rate variability, and then you compare, okay? And you see if there were changes or not across the intervention. But what you can observe is that your reliability drops, okay, when you do that analysis. So in this particular study, for example, we use vastus medialis, and then we check, for example, for recruitment threshold. We have a much higher ICC, a much lower SEM for the track model units compared to the classic averaging approach. And then the same thing for mean discharge rate and conduction velocity. But also, besides the increased reliability, we also have an increased sensitivity, which is very important for example, for interventions, where you really would like to see effects on motor unit properties. So then we took this data and we did an endurance training intervention, basically an intervention that we know that changes conduction velocity without changing the size of the fibers, okay? And we could observe that when we used the track model units, we had a much larger effect size, okay, on these changes in motor and conduction velocity pre-post sessions compared to the classic averaging approach when you just average one pool of motor units pre and post interventions. So that's one of the advantages of using this analysis. So then we applied, of course, this, it's important to apply these things in real training interventions. So in this particular case, we did after validating the approach, we did a high intensity interval training intervention, presuming that force would increase after that intervention. So it's a very short intervention of 2 weeks. And we could observe that force increased roughly about by 8%. And that also made the firing rate of these model units increase. So here again, we have 3 model units. I'm just depicting the first one. So that's the motor unit action potential profile for model unit number 1 here. And then we matched it with the post session. So this was 3 weeks apart. And then we have the action potentials for the same model unit after we match them by cross correlation, okay? So if you can see the action potential profile from pre and post sessions, they look very similar. And then by applying this matching, we can then quantify the model unit properties. And here we could see that the mean discharge rate was increased after the intervention. And that was related, of course, with increasing muscle strength pre-post intervention. Okay, so it's a very simple analysis, but it's very useful if you would like to analyze motor unit properties after interventions. But it's not that trivial, okay? And there's some things beyond the signal processing that are really important to apply, okay? These are photos from exact same study, okay? So we did two approaches, of course. The classic one is that you just mark the skin of the participants. But as you can imagine, these marks can, of course, go away. So we then also use transparent sheets as well to try to mark where the electrodes would be and use also anatomical guidance as well. It's really important to apply these things because as soon as, if you don't apply the grid in a similar location across sessions, then it's very likely that you won't have any clear matching between model units, okay? And it might sound really trivial, but it's one of the most important things to do and apply when you do any intervention for any muscle that you would like to assess. And how to do it? So how do we do this? So once you have applied like the electrode grids in the same position across sessions, and you are certain that you have performed all the measurements in a standardized way, the first thing, there are many overlaps with what you heard from Andrea as well, okay, with the conduction velocity. So the first thing, you have to spike trigger average your signal, so you get a clear view of propagation of action potentials, okay? Usually you have to select a window that you would like to see your action potentials, which is usually at established at 50 milliseconds. That allows a clear propagation view. Then after you have done your spike trigger averaging and you get your motor reaction potential profiles in a space, you can apply, let's say, a similarity measure for these signals. So the classic one to apply is cross-correlation analysis, but we have also employed other similarity techniques such as Euclidean distance as well. So it's up to you. For the purpose, of course, of openhdemg, we use cross-correlation analysis, but you could also use other similarity analysis. Okay. After you apply that analysis, you need to set a threshold, of course. We usually recommend a threshold higher than 0.8. And why is that? It's because the higher the threshold, the higher the similarity. Okay. So if the threshold is low, you are not very confident that you have an accurate matching. So that's the importance of establishing a really high threshold. And one important thing that I also forgot to mention in the first point is that this spike triggered averaging should be done in single differential mode. Okay, and why is that? In this case, it's not so much because you would like to assess conduction velocities and so forth, but it's because when you apply monopolar signals, you cannot see propagation of those action potentials. So then in single differential, the action potentials become more unique, and then it's easier for you to distinguish between different matchings, okay? So that's an important thing, because one thing that happens usually with people that is applying tracking, sometimes they track in monopolar mode, but the problem of that, you will have higher cross-correlation coefficients, but the problem is that also the different model units might look alike, okay? So that's an important step as well, to do the single differentiation, and then you apply the matching. Okay, but also one important step besides applying cross-correlation and besides applying thresholds is to visually inspect similarities between channels. Okay, it's not only, so never trust just the ICC result. Okay, so always look at all the channels that you have and then check if the template matches well or not. Okay. And also check the firing activity of the motor unit, which is something that we are going to see when we go to the openhdemg code. And also you need to be cautious with a few things as well. So when you track across different force levels and fatigue. So here I have an example for fatigue. So here we have tracked the same motor unit at the beginning and end of a time to task failure contraction. So, okay, the subjects here contracted until they could no longer contract, okay? And what you can see is the same motor unit, but at the end, so at the beginning is in blue and the end is red. You can clearly see that the action potentials increased in size, okay? And also they became wider, okay? And that also will affect when you track something, because if you, for example, want to track and the force has increased too much, then it's likely that even for a motor unit that might have been the same, will have a larger action potentials and also like extended action potentials as well that might affect the tracking, okay? So that's why it's important to check the whole grid to check how those action potentials are distributed to try to establish similarities. That's the reason why you need to check, for example, if interventions induce substantial changes in maximal force. So, for example, I think the longest study has been 4 weeks of resistance training in teres anterior muscle by Delbecq and colleagues, but we have no data from longer interventions with the tracking. So, and we have no data in muscles that can change in size as well, okay? And so it will be very useful to know, for example, if you have like a very long intervention that, let's say, lasted 3 months, if that tracking will be feasible or not. It's likely that the action potential morphology will change, and then, of course, the tracking might be more difficult to achieve. Okay, so now we're going to the practical part. So,
Which one do you want?
so with the within session first, but I can run
Let's exit.
because it's in the back mode. No?
Now it will exit.
That one.
So it takes time.
Okay. So well, the guys have implemented this code that we, of course, showed in Journal of Physiology in 2017. Again in openhdemg. So here at the beginning of the code, you can see how the functions, of course, and the files later on are imported. And again, I mentioned that there was some overlap with what Andrea was showing. So then you perform some filtering on the EMG signals, and then you arrange the electrogrid at the end, okay? And after doing that arrangement of the electrogrid, which is very important, then of course you will do the cross-correlation matching. One important thing, because I know that sometimes in certain sessions you might miss some channels, okay? It's important to remove channels, for example, if you have missing channels, because that can, of course, affect the cross-correlation measures. Okay, so this example, so we apply the tracking. I believe this contraction at 25% of MVC.
And this is 25.
Yeah, 25% in the vastus lateralis muscle. Okay. And here, so once you apply the tracking, on the top left, you have all the motor units that you have tracked, and then you can check all the possible motor unit pairs. So in this case, we have 10 motor unit pairs from 0 to 9. And then at the bottom left, you have the cross-correlation coefficients, and you have also the pairs that match, okay? So in the first case, so let's go, I will randomly apply it to the number 2. So here, for example, this is within a session, and you can clearly see that the cross-correlation coefficients are quite high, which is to be expected because, again, the electrode hasn't moved from the muscle, and therefore it's highly likely that you're going to get like an accurate match. And again, keep that in mind. So if you're tracking within a session, you can also think about using a higher threshold for your cross-correlation. But if you can see also the spatial profile, you can clearly see the action potentials are very, very similar between the motor units that were tracked across these 2 contractions. But also the firing properties for this motor unit look very similar as well. And that's what I was mentioning previously in the theory part, that you should check, yes, check the templates visually, but also check that the firing is correct. Because sometimes it can happen that, for example, the decomposition has identified a motor unit at the beginning and then lost the motor unit later on, and then used, let's say, less triggers to perform this spike trigger averaging for the template. And then you might have like an accurate tracking, like a matching, sorry, but the motor units are not the same, okay? And that's the reason why you need to be very conservative. Sometimes you will pass the threshold, okay? But if you see that the firing properties are very, very dissimilar, And also you're not very confident on the template matching, even if you have a high cross correlation, maybe reject that matching, okay? That's the reason why you need to do this. It's the same with all the analysis that the guys have shown, okay? With conduction velocity, with PICs, even discharge properties. You need to do this many, many times before you start like being confident in doing this, okay? But again, within a session, it's very easy. You can clearly see that almost all the model units match well. So if I go to model unit 6, for example, again, it's almost perfect matching. You cannot really tell what is the template 1 and 2, okay? But things get more complicated, of course, when you go to, I just close here. And then I run this. But things get more complicated, of course, when you start matching between sessions. And by the way, there are other ways to track. Of course, there are new methods coming up as well, and also you can match by filtering as well. But for openhdemg, we're just using for now the template matching that you're seeing now. So let's wait for a bit to track these files. So here we have the matching. Okay. So here, for example, this is a really good example. And by the way, you will always get less matching when you're tracking across sessions. And one important thing also to mention, because what you saw in the previous slides, where I showed that you have higher reliability. What is interesting, you have even higher reliability by having less model units analyzed. Because when you match, of course, you won't be able to match the whole pool of model units that you have identified, okay? But even with the few units that you identify, your analysis is also more accurate as well, okay? But in this case, for example, here we have 4 model units matched. Again, we have some cross-correlation coefficients which are lower, okay? And here it's when your expertise and of course your training will be very important. So here, for example, the first one has a cross correlation of 0.075, okay? And then if you start looking at on the distance, it might look similar to you, okay? But here are some parts of the grid which actually don't match quite well. And then if you go to the firing profiles, also there's a large variation in discharge rate, okay? Although if you do an intervention, you might find difference in discharge rate. It's to be expected, okay? But sometimes when you have this matching that you're not very confident, okay, or somewhat borderline, it's better to discard, okay? And try to be always using matchings that are higher than 0.8. So for example, now I'll go to the matching that is 0.8, which is this next one, and look at both the template and also the firing properties. And now you can clearly see. So here, for example, of course, do not expect the templates to be exactly the same because they will never be. Remember, you are removing the electrode and then placing it again, okay? Even if you're super accurate with that, it's likely that you will have some shifts and drifts in the signals, okay? Nevertheless, in this case, the profile and representation of these action potentials are very, you can clearly see that are similar, but also if you see the firing properties, they look more reasonable as well between both model units. And one is important as well, that I didn't mention in my previous slide, be careful with the positioning of the limb as well, okay? Because if you measure at different lengths, it's likely that you will get different spatial profiles as well, okay? So if you track, please try to track within the same position so you don't have issues with that as well, okay? And then lastly, I will check the last one, for example, 0.71, which is the matching number 3, and you can clearly see that they don't match at all. So for example, here, you can clearly see that, first of all, the STA doesn't look as nice as the other ones, which this one, as Andrea was showing, doesn't have very clear propagation compared to the previous ones. And also you can clearly see that the firing properties are very different as well, okay? That's it.
Okay, thank you. Thanks a lot, Eduardo. Thanks a lot, everybody. We are out of time, but thankfully the next a workshop allowed us for 5 more minutes. So I will invite on the stage Francesco and Gregory. They decided to join the project, and so they can give us a feedback on why they think it's worth doing so.
Thank you. So let's begin with the questions, since we are a little bit out of time, and maybe we finish in the comments. Not so many questions.
Yeah, we will be around all the time to ask any question.
I can maybe try to comment a little bit what we have seen today. So all the analysis that you have seen today in this very nice tool that Giacomo and others have developed, obviously comes after the composition in T. So then you need to see how confident you are on your decomposition results and on your cleaning procedure that you have performed on the remote. One thing that I want to point it out is that all the measures that you have seen today, there are some measures that can be more sensitive to errors in the decomposition and measures that are less sensitive. So based on that, you need to judge the quality and let's say, which unit you are going to use for this kind of analysis later on. For example, just to start from let's say the first presentation, the mean discharge rate in the plateau, usually not so sensitive to errors in the decomposition unless obviously the module is a bit better and say decomposed. On the other hand, the coefficient of variation in interfatigue is very sensitive. So once you have if you have a unit where there are a few spikes or a number of spikes that you are not sure 100%, if you want to report the mean discharge rate, would be okay. If you want to report the coefficient of variation in the spike interval, you need to have, because that's will be a bias. Other measures like spike trigger average, not very sensitive or not extremely sensitive to errors. So then in that case, even if you are not sure about if you fire in your local unit, you will be able still to use spike trigger average. On the other hand, that type of measure at the beginning and at the end of the rank, you need to be sure 100% that your spike have a reasonable. Otherwise, you will get into trouble. Just wanted to say that because it's important, and each of us will face that problem working on our unit, and so I think this is important to say. Keep in mind that depending on the measure that you want to apply, then obviously you can judge the quality, let's say, the results of the tool.
Thanks a lot, Francesco.
Yeah, I just want to point to the importance of reproducibility and how open science helps contribute to that. We started doing some of these persistent recurring estimations in real motor neurons a few years ago. And what we quickly realized is that you can change one parameter and find the result that you want to find. And then if one person is saying they found something and then someone else does the same experiment and they didn't find something, you quickly get into either arguing between groups or just having a lack of confidence in a method altogether. And so at the very least, if you want to try something new, repeating what someone else has done is a nice way to show that, well, if you do it the exact same way, there is still a difference, but we think this new way we're going to approach something might be better. We do it in addition. And so having these codes open access for everyone to adopt is one way that we can facilitate that. So it's really easy to do these analyses. You don't need to write your own code to replicate them. I think that's really important for science moving forward. And I think this whole initiative is really important for that reason. And just in the interest of time here, I won't blab on about it for too long, but immediately I was a little hesitant to, throw code around for people to just analyze things as they wish, but really jumped on board. I think it's a really important thing that people use similar codes so that we can make sure that if we're saying someone didn't find something versus we did, we know that the reason is more than just a 5 instead of a 10 that slipped into one line of your code. So hats off to these folks for making this available to.
Many thanks, many thanks. Okay, if you are using the framework, please cite the article, cite also the articles of those who developed the analysis techniques, and please leave a star to our GitHub repository because this is a huge help for code developers. Thanks a lot for joining today.